Hello, everyone. Hello from us, from MaxWell Biosystem. I see more and more people are connecting, and this is great. We will give maybe a couple of more minutes for more people to join. I'm very happy to actually host back a webinar after a long time. We have a very packed season before the end of the year. And I'm not doing it for a long time, so happy to be back. I'm also very happy to have our guest speaker for the day. We have Dr. Xuan Lin from the Center for Genomic Regulation from Barcelona, Spain, and also Philip Ornauer, who is also a colleague of us working with us at MaxWell now. Do you guys want to say something to our audience while we have more and more people joining? You are from Barcelona, Xuan, lucky you now.
Yes. It's been nice here in Barcelona, very sunny. Hi to everyone who's still joining. It's very nice. Thanks for having me here. Super happy and looking forward to the webinar.
Our pleasure. Philip, also welcome to your first webinar.
Yeah, thank you so much. I'm glad that I can be part of this. Glad that we can present our joint work. It was really great experience and happy to share it with everyone.
That's great. Indeed, as I said, we have a very packed list of webinars coming up before the end of the year, but this one in particular is going to be very relevant for people interested in the neuronal programming. And so you will have the chance to ask more and more questions to our speaker after their talk. So for the people that are not familiar with us, we are actually a company leader in the next generation electrophysiology. Our products are mainly one, Max1 and Max2. And what is important is that not only we provide hardware solution, but also software solution and application solution. And here we actually have a hub that serves all of our users. Our users are located in over 20 countries, 23 at the moment that we can count. So if you want to add more, we are more than happy. And our technology is actually based on our background, our backup in R&D is over 20 years. And this helps us to be leader, to have a very high reputation, top reputation in the scientific community. Thanks to our solution that I said are going from the hardware, software, and application. Actually now our users published over 200 articles in peer-reviewed journal. We have now over 200 publications, as I said, and these are just a few snapshots of relevant publication. If you're interested and you want to know more, because we couldn't clearly fit all in a slide, you can look at our website or directly scan the QR code that is here present, and then go directly into diving into the publication. I'm gonna ask for help. Can I switch? Okay. At MaxWell, we also have a motto that is every cell has a story to tell. But then the question is how we're able actually to understand the story of each cell. In the past, to have information about the functionality or even morphology of a cell, what we could do, we could actually use different techniques from the standard immunofluorescence imaging or the gold standard in electrophysiology that was the patch clamp. Both of them are still in use, but with them, there are also some limitations. For example, if you want to do imaging, then you are kind of forced to do endpoint experiment because you would have to use label together with your sample. In case of patch clamp, instead, you can only look at the activity of a limited number of samples. And this is also a pretty invasive technique. With our technology instead, which is the high-density microelectrode array, we actually provide a solution that combines both techniques. You can have information about the electrical signal, but you can also have information about the morphology, exactly as is shown here in the middle in the video. So now you can think about, okay, how can I imagine my example? If you think that your sample, your culture will be on top of our chip, the type of information that you can get are about the spiking activity. So for example, firing rate, spike amplitude. You can get information about where are located the active cells by looking at the activity map, and also information about the network dynamics in your sample. So we know that when you do your research, it's important for you to have the perfect experimental success, the one that maximizes, as I said, your possibility of success. So there are some key elements that are also very important, and this is what we focus about when we try to provide the best solution for you. Because of course, we design our hardware, our software, so that you can have a comprehensive overview of the neuronal characterization of your sample, so that you can do experiment and get high quality data, reproducible data, consistent results. Very importantly, also all of these, we have made it very simple for you. Our technology is very easy for you, user-friendly. I was a user myself, so I can confirm that. And on top of that, it's very important for us to provide you a solution that will allow you to advance beyond what is the current state of art, so that you can go beyond the current boundaries in the neuroscience research. So, as I said, it's important to be able to read the story of every cell. How you can do that? If you're working with a low-density MEA, there is the problem, because then in case your cells is not sitting on top of a limited number of electrodes, then you are going to miss out on a signal. While instead with high-density MEA, and in particular with our platform, we have over 26,400 electrodes per well with a very short pitch distance. The center to center from an electrode, it's just 17.5 micrometer. And this is important so that in this way you are sure that you are going to record actually from every cell having multiple electrodes available just for one cell. And if you're used to work with the iPSC-derived neurons, or for example with organoids, you also know that sometimes the action potential, the signal that you're working with, is very small. In particular, when the signal, for example, propagate from the action initial segment throughout the axon. And even if you're working with some other high-density MEA, maybe in that case, you might be fighting with a very high background noise, and this background noise can actually mask the signal that instead you are interested in. In that case, instead, with our solution, you don't have to be worried about that, because we made it super easy for you, also with our low noise readout that will allow you to record even the smallest signal with very high fidelity to have only high quality results. An example of this is the axon tracking assay. In this case, this is actually an assay that we developed. It's our unique assay, and will allow you to get information about the morphology and also the functionality of the cells, because we can actually reconstruct the footprint of the actional branches. So to look at how the signal propagate from the action initial segment throughout the branches. You can see it in the footprint image on the right side of the screen, but also in the video that was actually replaying on the left side of the screen. So once again, our technology is based on a very large area chip, 2 by 4 millimeter sensor array area with 26,400 electrodes, with very low noise readouts. And importantly, you have the perfect condition for your experiment, because you also have flexible electrical stimulation as a possibility. All of these electrodes can be eventually stimulated up to 32 simultaneously together. And what we are very proud of is that we didn't decrease the number of electrodes when instead we increased the number of wells. All of our consumables, from the single well to the 6-well plate to the 24-well plate, have 26,400 electrodes per well. And in this reason, you don't have to be worried about sacrificing some of your experiments, sacrificing on the output that you can get, but actually, while instead you want to go out with a number of wells. Our consumables, so the chip or the plates, are compatible with these two systems, as shown there, MaxOne and MaxTwo. These are the two platforms that we're working with, and they serve different scope. The MaxOne is what we consider a flexible platform because it's way smaller, more or less the size of a phone, and it can be used for culture, also for advanced application, because you can combine it, for example, with microscope, perfusion system, depending on what you're interested in. While instead the MaxTwo, this can be used for long-term study and is in particularly suitable your research in case instead you are more interested to have a higher throughput, because indeed it is compatible with 6 and 24 well plate. Not only we serve the perfect hardware solution, but also we provide a software. And we want to consider, like to consider our software in all-in-one solution, because with MaxLab Live, you are able to visualize, record, analyze, but also immediately visualize the metrics and the report of the data that you actually recorded. In our software, we have 4 different assays. The activity scan assay is the one that will allow you to visualize where are the active cells, where they are located in the samples. The network assay is inside the assay to look at how cells are connected, how they are synchronized, what is the network dynamic in your sample. The axon tracking assay, I described it before. This is our unique assay. We are the only one who are actually able to trace the axon excitability and also to look at the axon morphology. And then with the stimulation assay, you can also look at cell excitability because you can evoke basically a response after certain stimuli. At this point, you might wonder what our system can be used with, what type of samples. It can be used for 2D samples in case of iPSC-derived neurons, in case of 3D samples like organoids, spheroids, but also primary neurons, I forgot. Other type of samples can be retinal brain slices, but also you can use it with other advanced applications like in neurocomputing or with microphysiological system. In these slides, we have just a few examples of some data that were actually collected by some of our users, just to show you that our system and our assay were used with a variety of 2D neuronal culture, from primary neurons to motor neurons, dopaminergic neurons, GABAergic, glutamatergic, and the list can go on. But now there is a question, and now that is like, okay, but if I have so many cells, how can I actually distinguish between the different network signature of the different cells? And actually, I would now call my colleague Philip to address this type of question. And why? Because I think that with his background, he would have the perfect expertise to actually address the next point. Just to give you an introduction about Philip. Philip is a software engineer and computational neuroscientist at MaxWell Biosystems. And so with his work, he's also responsible for the integration and development of all of these data analysis algorithm inside MaxLab Live. He received his PhD in neuroscience from ETH Zurich, where he also stayed for a postdoc. And so, as I said, he's the perfect person actually to guide you through the next steps. And Philip, the stage is yours now. Thank you.
Thank you so much, Laura, for the very kind introduction. So yeah, I'm going to give a very short overview of what you can do after the recording's done with MaxLab Live before we jump in right into the main story. So one of the key components here, or the 2 main components here that I'm going to briefly mention, are Spike Interface and DFIS. So Spike Interface is an open source tool that's Python-based, which is a spike sorting framework. So spike sorting, in case you're unfamiliar with that, refers to the analysis where we can really extract single cell information from the recordings. So this is really a highly delicate process, and it's really only possible due to the very short electrode pitch and the high signal to noise ratio of our devices. And after the spike sorting, you have the single cell information, and DFIS is a tool that I developed as part of my PhD project, and we're now trying to integrate parts of that into the MaxLab Live framework that is MATLAB based and allows the functional phenotyping of these individual neurons, but not only on the neuronal level, but also on the whole network level. So I'll briefly give you a very high level overview. I'm going to show more of the actual data later on after the genomics introduction by Sichuan. So the key component of this whole toolbox is really the feature extraction and integration. So what does that mean? While single cell activity itself is very informative, it's also very high, like a highly complex data set. So what DFIS does is that it really allows you to look at features on a very detailed level, on the waveform level, which means the exponential shapes, which might be indicative of different cell types, the activity features, meaning how does the neuron fire? Is it more bursty? Is it non-bursty? All of these can also, again, be informative regarding cell types or dysfunctions. And then we come to the network features where you can look at burst dynamics, but also at connectivity features, which quantify how the neurons communicate with each other. So here, for example, you can see the output of the spike sorting on the left. So you really see how many electrodes actually cover one single neuron. And on the right, you see the features that we can infer from this exponential waveform, as we call it. And on the other hand, on the, like, let's say, opposite side of the scale, we have these network features where we really look into the connectivity of the network, but also like how the burst shapes form over time. And those can be typically very informative about the maturity and the strength of the connections within these networks.
Thanks a lot for having me here. As Laura introduced, I recently started my group at CRG in Barcelona. So for today's presentation, I'll mainly talk about my postdoc work, which is carried out in Barbara Teutlin's group in ETH Zurich, where we start from iPS cells to induce them into different neuronal subtypes and use single-cell genomics as well as electrophysiology to profile their diversity and sort out potential regulatory mechanisms. All right, so human cells can be engineered in vitro from iPS cells into all kinds of cell types. And since the iPS cells can be directly reprogrammed from fibroblasts of each individual, these in vitro engineered human cells and tissues can serve as a powerful tool for modeling human biology and diseases. Now, this also is very promising, but this, of course, is a rather simplified scenario because real human cells and tissue are really diverse. Take the human nervous system as an example. So the human nervous system is diverse. It composes of at least two divisions, the central nervous system and the peripheral nervous system. And in the central nervous system, there are different, it contains the brain and the spinal cord, and the brain has different regions in it. And in different, even in the same brain regions, neuron can secrete different neurotransmitters. So this makes engineering human cell subtypes in vitro looking like this. It's quite challenging also, but the central question here is how do we know, like how similar are these in vitroly engineered cells and tissue compared to the real human cells and tissue? So single-cell transcriptomics can come in as a powerful approach here because there's a lot of human cells and tissue atlas out there. So with this reference atlas, we can assess the quality of iPS-derived cells by really to look into the composition of these cells to see if there's any off-target cells and to directly quantify the similarity to real human cells and tissues. So this is exactly what I do in my postdoc, and you can already see here, I'll start with quite some genomics analysis before we really go into the electrophysiology part. So in my postdoc, I've been working on a model system, which is a neuron that are directly induced from iPS cells by simply overexpressing one transcription factor, neurogenic 2. The resulting neurogenic 2 induced neurons were abbreviated as NG-into-I neuron in this presentation. I'm especially fascinated by the system because NG-into-I neurons are induced with extremely high efficiency and mature way faster compared to in vivo. So NG-into-I neurons, since it's so powerful, has been widely used for drug screening and disease modeling, and it was believed to be purely cortical excitatory neurons. But at that time, no one really systematically assessed the identity of NG-into-I neurons. So together with Justin and Sebastian, we took the chance and used single-cell RNA sequencing to profile the identity of NG-into-I neurons. we take time point of engine-to-eye neuron differentiation, and you can see the engine-to-eye neuron we start from iPS cells, so we got pluripotency markers at the beginning with NANOC, and it progressed into some intermediate states, followed by expression of neuronal marker MAP2 starting from day 5. But what we immediately notice is that engine-to-eye neurons is actually molecularly heterogeneous, which composed of at least 3 neuronal fates. And these neuronal fates are marked by more central nervous system markers, GPM6A, and more peripheral nervous system marker, peripherin. And it can be then further separated into FOX2B positive and POL4F1 positive neurons. So engine-to-eye neurons are not only molecularly heterogeneous, but it's also quite confused. It has a fate of a combination of CNS and PNS. So this is quite surprising to people at that time, but if we look at the expression pattern of NGN2 in early neural development, this heterogeneity of NGN2-eye neuron is not super surprising anymore, because NGN2 is actually widely expressed across the nervous system. So we start to think about the possibility of combining NGN into expression with the morphogens. They are involved in patterning and regionalization of early neural tube developments to see if we can guide NGN2-eye neuron into associated lineages. So the morphogens, on the right-hand side, you can see a developing neural tube. With the morphogen we consider here, establish the anterior, posterior, and dorsal ventral axis. For example, the Wnt signaling gradients, which can be modulated by Wnt agonist chirin or Wnt antagonist XAV, are important for establishing the anterior posterior axis. Retinoic acid is really important in shaping the hindbrain and spinal cord sulfate, while the FGF8 is involved in most anterior part of the forebrain and mid-hindbrain boundaries. Well, for the dorsal and ventral patterning morphogen, we're more interested in BMP4 and sonic hedgehog. So with all these morphogen modulators that I just mentioned, we started a combinatorial patterning screen of engine-to-eye neuron to see if we can guide them into associated lineage. We start from iPS cells, induce engine-to expression with doxycycline, followed by anterior, posterior, and dorsal ventral patterning. And at day 10, the resulting patterned engine-to-eye neurons includes 192 morphogen gradient combinations are harvested for highly multiplexed single nuclear RNA sequencing. So together with Jesper, we start to look at the data, and among the 192 morphogen gradients combinations, we're able to profile nearly 200,000 cells and identify 101 molecularly distinct clusters. And with the control cell here, you can already see how combinatorial patterning can expand the diversity of engine-to-eye neuron subtypes to the space they are not seen in control before. And with here you can see how each anterior, posterior, and dorsal ventral morphogen contributes to this diversity. We see a very interesting, nearly mutually exclusive but mirrored cell fates that are generated by dorsal and ventral morphogen, BMP4 and sonic hedgehog. So we wanted to dive deeper and look into how morphogen gradients shape this the engine-to-eye neuron diversity. So on the left-hand side, you can see retinoic acid gradients and the resulting cell fate. You can see a rather discrete change of cell fate once we increase retinoic acid gradients. And at highest retinoic acid gradients, highest retinoic acid concentration, the cell fates that it generates is more homogeneous. But on the right-hand side, in contrast, you can see a chirin-mediated cell fate based on different chirin gradients. And here you can see rather continuous change of cell fate instead of the discrete one that we see in retinoic acid. And highest concentration of chirin did not give rise to more homogeneous cell fate. So this morphogen gradient dynamics is really morphogen dependent, and higher concentration does not necessarily mean more homogeneous cell fate. We wanted to know what are the neurons that we patterned in the dish. So we annotate the identity of these neurons by marker genes as well as comparison to primary reference atlas. And the final annotation is based on their division, the region, and the neuron type. So patterned NGN2I neuron have both CNS and PNS neurons. And in terms of regional identity, it's quite diverse. It has like forebrain, midbrain, hindbrain, and spinal cord identities as those in the CNS, but it also has more peripheral nervous system identity, such as those in the sympathetic nervous system and enteric nervous system, trigeminal ganglia, and dorsal root ganglia. And in terms of neuron type, we got a majority of glutamatergic neurons, which is in line with the known identity of NGN2I neurons, but there are also quite some cholinergic and noradrenergic neuronal fate. We want to know how morphogen combinations give rise or contributes to this diversity and what are the underlying mechanisms. So what we do is that we infer a gene regulatory network that are centered on morphogens. In this gene regulatory network, you can see that the morphogen is at the center, while each node represents a regulon, meaning transcription factor and their target genes. The number of the size of the nodes represent the number of target genes, while we color the node based on the regional identity that has highest corresponding regulon activity. So based on this analysis, we can see that morphogen combination can activate distinct regulon network to shape the diversity of pattern NGN2I neuron. For example, retinoic acids can control RARB and HuxB4 regulons, which is then linked to more hindbrain identity, while the BMP4 is associated with FOX2B and SMART regulon, which is linked to the more peripheral nervous system identities. But these are inference analysis, and we wonder whether these, like how much does these regulons contribute to the associated identity, whether these regulons are necessary for the identity, or more boldly, whether these regulons are sufficient to drive the corresponding cell identity. So together with Benedict, we try to validate the inferred gene regulatory network by analyzing the necessity and sufficiency of regulon hub transcription factor in driving neuronal identity. The relationship we have now is how morphogen is linked to a regulon and how it's linked to neuron subtypes. If the regulons are necessary for the corresponding neuron subtype identity, once we knock out the regulons, even under the presence of morphogen, we won't be able to achieve the link subtypes. Well, while if the regulons are sufficient, we just need to overexpress the regulon to achieve the neuron subtype without the need of morphogens. So this is exactly what we do. And I'm showing you the results of the knockouts or the necessity experiments. While the top panel, I'm showing you 4 regulon activity from 4 transcription factors at the regulon hub, while the lower panel is the cell density of knockout regulon presence compared to the control. What we basically see here is that once we knock out regulon hub transcription factors, we won't be able to achieve the cell fate where the regulon activity is high. This means that these inferred regulons are actually necessary, and without the regulon, we won't be able to achieve the linked cell fate. But on the other hand, if we overexpress the regulon hub TF transcription factors in addition to neurogenin-2, we're able to push the control cell fate, as shown on the left-hand side, to the cell fate where the regulon activity is high. This suggests that the inferred regulons are sufficient to guide the specification of neuronal cell fates, independent of patterning cues, because we didn't use any morphogen here. And what's more interesting for us is that the purity of these cell fates result from overexpressing regulon hub transcription factors seems to be more pure, at least in certain conditions, compared to the patterning screen, which suggests that overexpressing transcription factors can serve as a strategy to engineer more homogeneous and more specific cell subtypes. Now, with all these molecularly distinct neuronal cell fates, we wonder whether they are functionally distinct and whether they are functional at all and whether they are functionally distinct. So I'm going to hand over my presentation to Philip so he can tell us more about the electrophysiology part of this talk.
So I'm going to talk about the functional assessment of those patterned NGN2s, which I did together with some of my colleagues back at ETH. And yeah. Let's see if I can control the slides. Yes. So we used both the 6 and the 24 well plate with the MaxTwo system. And yeah, something that we already quickly mentioned earlier, we extracted a whole lot of different features on like the whole area of scales from the spike sorting results, the footprints. We looked at the network activity. We looked at the axonal tracings, which you can see here, which we then used to infer the functional morphology, which we then also compared between the different conditions. So yeah, so this is the overview. We looked at the single neurons, the network characteristics, and the axonal morphology, and I'm going to go through them one by one, starting with the single neuron activities. So what you can clearly see already is here in these spike trains, which corresponds to different action potentials by each of those neurons. And you can clearly see that we have very distinct activity patterns on the single cell level. And if we, similar to what the RNA sequencing data shows, throw them together into a UMAP and a clustering analysis, we see very distinct clusters. And most importantly, we see very homogeneous clusters between cultures of the same condition. So this graph is a bit more complex to interpret, so I'm gonna spend a bit more time. So what we did here is we really compared the composition of one network. So each of those dots or each line corresponds to one network, and we inferred the composition from those single neuron clusters, and we compared them to all the other networks. And what you can clearly see, for example, here in this condition, the RA and BMP4 one, we see a very high homogeneity between the different networks. So this gives us a very good understanding of the single cell composition, and it also tells us that the composition from the patterning really results in very homogeneous compositions. But it also tells us, for example, that conditions can be very distinct. So this also goes in line with what Sichuan mentioned earlier, is that you can get very distinct single cell compositions. So this was already very encouraging. However, neuronal activity goes beyond simple single cell activity, and we're also very interested in the network. So this is what we did next. And across even 3 different batches, so 3 different differentiations, we inferred the network phenotype. So here we looked really at a wide range of features. So this is kind of part of the DFIS toolbox. So here you can really look at connectivity features, at burst metrics. We can look at firing patterns. And all of this combined can be, again, thrown into the UMAP dimensionality reduction just to highlight the similarity. And we can also kind of see a gradient between the RA, which is the orange and the blue on the top left, and the GER, which is in the bottom right. So we can clearly see that there's a gradient from the RA to the GER, which is super interesting. And even, again, as I mentioned initially, between the different batches, we see a very consistent network phenotype. So we also were interested in which of the features actually were most predictive of the underlying patterning. And we found that, interestingly, there were, like across all the scales, we found different features that were important. So we found, for example, that the network density, so the amount of connections that the neurons form, is very different, and also the network efficiency. So, for example, the RA plus BMP4 condition has a very high network efficiency and a very low network density, while for the CHR one, it's the exact opposite. So this, for example, tells us that the CHR condition gives, maybe it doesn't have the synaptic pruning that we associate with more mature neural networks. But also, so the action potential rising phase is different. So this also tells us that even on the single cell channel kinetics level, there's differences, and then very standard features like firing rates are also very predictive. So this was nice to see that really the whole spectrum of features that we're looking at gives us information about the underlying patterning. And then, in my opinion, at least, the most visually appealing one is the functional morphology. So as Laura mentioned initially, really the only hardware currently capable of inferring these functional footprints, and those gave us really, really nicely separated functional footprints for each of those neurons. So you can see again, like this gradient going from CHR, giving us very small, very immature looking neurons and axons, going to the RA conditions where we have really, really far-reaching axonal protrusions. And if we compare those using Scholl analysis, where we can systematically compare different features of this axonal outgrowth, we can see that, for example, the number of intersections and the branching index, again, like CHR ranks very low on both of them. And the RA condition, for example, here in orange, ranks very highly. And again, if we put all these features. So this is only a subset of the features that we actually looked at. If we, again, use UMAP to really show the clustering of those different conditions, we see again that on the top right, we have the CHR conditions clustering together. On the bottom left, we have the RA conditions with the BMP somewhat separate from there. And yeah, so this tells us that even using the functional morphology, we can kind of distinguish between the different patternings. And this is, again, as Laura mentioned, most importantly, without labeling. So you can really also, in theory, you could look at this in a longitudinal way. You could look at this at these footprints on different time points. And so this is, yeah, I mean, as nice as it gets if you see the final result. So here we correlated the transcriptome maturation score. So this is really an aggregated score where we that is comprised of synaptic markers, morphological markers, and all that. And we correlate the so this is from single cell RNA sequencing. And if we correlate this with the shoal features that we're looking on the left, then we basically have almost a perfect correlation between those features and the genetics. So this gives you really an impression of how consistent this phenotype really is, and with how many different ways we can functionally assess these different patterned conditions. Yeah, and with that, I'm gonna hand back over to Xuan.
Thanks, Philip. Okay, I think I have. Okay, so we've been showing you neurons, NGN2I neurons that are post-pattern, meaning patterning after NGN2I neuron overexpression starting from iPS cells. But in real neuronal development, patterning and regionalization actually happens before neuronal differentiation. So we start to wonder what will happen if we first pattern the iPS cells into diverse pre-patterned neural stem cells and progenitors, and only after that induce NGN2 expression. And how does NGN2 really interact with these diverse input cell states to generate different neuronal subtypes, and whether these subtypes are different from the post-pattern neuron I just mentioned. So this is called a pre-patterning screen because patterning happened prior to NGN2I neuron, prior to NGN2 expression. Yes, so with this pre-patterning, we're able to get really diverse neuronal progenitors, including those that are shown here at this pre-patterning stage. Here we have the multiome data, meaning we have single-cell transcriptome and single-cell open chromatin data, so that we can look into how epigenetic landscape how does NGN2 interact with these diverse epigenetic landscape to induce the resulting neuronal fate? So since we're especially interested in NGN2 binding to these, how it interact with the epigenetic landscape, we look specifically into the NGN2 motifs from the open chromatin data. So first, we look at the inaccessible NGN2 motifs and what are the potential genes that are linked with these motifs. So what you can see here is a bunch of synaptic genes and neural differentiation genes, which is not super surprising because NGN2 is known as proneural pioneering factors, meaning that it will be able to open up the chromatins that are linked to neuronal differentiation. What's more interesting to us is the accessible NGN2 motifs, which has largely enriched in patterning and regionalization. And many of them are differentially accessible in different regional specific neuroprogenitors. So now comes the question, how does NGN2 interpret this differential accessibility in different progenitors? And how does this interact? How does this interaction affect the resulting neuronal fate? There are different scenarios that we're considering. So first of all, this is the control scenario where we put overexpressed NGN2 on top of iPS cells, getting some control neurons. Well, if NGN2 is a general pioneering factor, regardless of the pre-pattern progenitor identity, it will be able to induce the similar control neurons, similar to those directly induced from iPS cells, even if we start from different pre-pattern progenitors. Alternatively, NGN2 might conform to the identity of these pre-pattern progenitors, which result in the neurons of corresponding regional identity similar to progenitors. Or NGN2 can interact with the chromatin landscape of progenitors to influence the resulting neuronal cell fates, which creates a set of neurons that have quite distinct regional identities compared to the progenitors, which is also different from the control. So since we have profiled the resulting pre-patterned NGN2I neurons using highly multiplex single nuclear RNA sequencing, we'll be able to answer this question by looking at the data directly. So our pre-patterned neurons include additional 96 patterning samples, and we profile more than 300,000 cells and identify 115 molecular distinct clusters. And you can see here the regional identities and cell types are quite different from the control, as well as the post-patterned NGN2I neurons. And to answer how NGN2 interact with diverse epigenetic landscape, we try to compare the regional identity in progenitors and the resulting NGN2-induced neurons. And you can see a very rather high correlation of regional identity composition, meaning that NGN2 does not actively involved in the regionalization of neurons, but rather directly conform to whatever chromatin landscape they are pre-established in the progenitors. So the NGN2 is not so much of a pine neuronal pioneering factor that we think of, but it rather have some restricted specificity in different input states. So we wanted to know how the pre and post pattern NGN2I neurons are compared. Are they the same or are they different? So what we can do is directly integrate these two data sets. And you can see that they occupy quite different space on the UMAP, meaning that there are distinct neuronal subtypes. And that pre-patterning can create NGN2I neurons. They are beyond the diversity of those of post-pattern NGN2I neurons. And this is because the underlying gene regulation is already different. We look at the gene regulatory network, and here you can focus on the color of the edge. You can see the pre and post patterning morphogen result in different regulon networks. And that the pre-patterned regulons are more like pre-patterning actually allow the acquisition of regulon network. They are more similar to the primary neurons. We also look more deeply into the maturity, heterogeneity, and similarity to primary neurons in terms of NGN2I neurons directly induced from iPS cells, were those induced from pre-patterned progenitors. So you can see that pre-patterned NGN2I neurons are less mature compared to the control iPS-induced neurons, and they're also less heterogeneous, meaning that they are more pure in a way, and they are more similar to primary neurons. So if people were to consider engineering neurons in vitro, one can consider using pre-patterned progenitor as a starting point, which can make it more homogeneous and similar to primary, just that it matures slower than directly induced from iPS cells. So we wanted to know whether our combinatorial patterning approach can serve as a more general strategy for inducing diverse neuronal subtypes or cell subtypes in general. So what we do is that we try the same approach, but now on induced GABAergic neurons from overexpressing ASCL1 and DRX2 transcription factors on iPS cells. And with additional 192 samples, we're able to generate diverse induced GABAergic neuron subtypes, similar to that of the induced NGN2I neurons, which suggests that this can serve as a general strategy. So we put together, with all the data that we generated, we put them together and in vitro engineered neuronal diversity atlas for the community, which includes different neuronal subtypes that we're able to generate, including their division, the region, and the neuron type, and the conditions we use to generate them, including transcription factors, timing, different morphogen concentrations, and the resulting purity of the specific subtype in each conditions. We also look into the enrichment of disease genes and ion channels just for people's information. Now, at the end of the day, we wonder what neurons are we really engineering and how are they compared to the primary neurons. So what we do here is we try to put together a systematic comparison with primary neurons. We collect 11 studies of primary neurons with different regions and neurotransmitter and try to project our patterned eye neurons on top. And based on the coverage you see here, it's kind of a bittersweet because on one hand, we're happy that we're able to generate such neuronal diversity in vitro in a single study. But on the other hand, there's also way more neurons, primary neurons that we haven't been able to reach in vitro. So this point to a direct next step that we can further expand that diversity of engineered neurons in the dish. Now we can dive deeper, and we're able to identify one-to-one correspondence between primary neurons and the engineered neurons based on gene signature scores. You can see here very nice correlation. This gene signature we're able to recapitulate. So for the top matched hits, we're able to find high correlation between the marker genes, transcription factors, ion channels, and synaptic genes, that we're able to recapitulate very nicely in vitro, but we weren't able to recapitulate the oxidative phosphorylation genes expression, meaning that the metabolism of in vitrally engineered neurons is quite different from those of in vivo. And this is also a directly next step for engineering more neural subtype that are more similar or more functional to compared with primary. So in summary, I've been showing you here that we combine proneural paraneuron transcription factors with morphogen gradient combinations to start with a combinatorial morphogen screen with 480 conditions and profile over 700,000 single cells to generate diverse neuron subtypes that are distinct in morphology, function, and their transcriptomes. You can see how important electrophysiology is in this context, because we really need to know if these neurons are functional or functionally distinct, and we can also look into their morphologies. We're also able to identify the regulatory network and cooperative signalings that are underlying neural fate specification. So with that, I would like to thank the quantitative developmental biology lab, especially the supervision from Barbara and Gray, and it was very nice to work together with Jasper, Jason, Sebastian, Benedict, and Anne-Sophie towards these projects. Thank our collaborators in electrophysiology, University of Bonn, for their support in induced GABAergic neurons, and Genomic Facility Basel for sequencing all our libraries. Here are the funding buddies, and thank you all for your attention. I'll be happy to take any questions.
Thank you so much, both of you, for this amazing talk, and also congratulations once again for your publication. It's a science one, was very recent. Very, very happy for you both. So I do see the first question already popping up in the QA, and now I will ask to all our attendees to don't be shy and actually start to write there your questions. I will come to them just in a second. Before that, I want actually to invite you to some of our next event, just to give you more time to write the question and think about them. We know it's the end of season, SFN, it's back, and we actually have a lot of events going around there. We are gonna not only attending the conference, but we also have a social event. It's on the night of November 18. You can directly register if you scan the QR code here on the left side of the screen. So in case you plan to attend this event, we are also looking forward to meet you there, not only at the booth, but also at the event. In case you are interested, there is still time also to apply to two things. One is the travel grant, actually, to attend SFN. There is still time, couple of days still left until October 21st. And then we have also a competition open for all of our MEA artists, because if you have anything, if you're already using our technology and you have any type of art that can be connected to our link, then feel free to submit your masterpiece directly using also the QR code, or you can also find more information on our website. So back to our speakers. Are you ready? Now it's your turn. So
Exciting.
Good. So the first question is from Camilla. Hi, Camilla. So she say, hi, thank you so much. Such a great paper. I was wondering how many background iPSCs derived from distinct individuals did you analyze, and what is the neuron variability without patterning?
So thanks for the question, Camilla. We there's a previous paper in 2021 where we use 2 additional lines, and we're able to show that the resulting neuronal variability is quite similar between lines, just the proportion varies a little bit. And in this current studies, we tried 2 additional lines as well, and results are quite comparable to previous. I hope this answers your questions.
Thank you, Suchan. And then we go from the next one from Neil. Did you find evidence for any of the canonical interneurons in your culture? And then he lists a bunch of markers for interneurons there, PV, VIP, any of those.
Yes, so we do have somatostatin positive or like SST positive interneurons in our cultures. For the other 2s, I am not quite sure at this point, but I can for sure check. Or the data is actually public, so if you have interest in looking into them, you can also look into that. But yes, so for sure SST positive.
Perfect, thank you. We'll jump into the 3rd one then from Amalia. There are reports on in vitro neuron having multiple AIS. How does EFI software account for such possibility? Does it choose the best guess for the axon, or does it allow for such possibility? And now maybe also Philip can jump in. Sichuan, up to you, how you want to address the question.
I think Philip will be the ideal person to answer this question.
Yeah, very good question. So we've seen also cases like this before. Typically, it's not really a big issue. So the biggest problem I would say is that this can kind of mess with your spike sorting. So in that regard, you might end up with the spike sorter or the analysis thinking that you're looking at 2 different neurons that then show very similar activity. And then kind of in a post-processing step, you can then go and look into the 2 activity patterns. And if you see, well, the overlap is basically 100%, then you can go back to saying, well, we merged these 2 neurons into one because we're very confident that this is only one. So this is kind of the way that I typically see this going in the analysis.
Thank you, Philip. And we actually have a second question from Amalia, so we'll just keep going. And is, how does the single-sex RNA-seq clusters map to electrophysiological activity? The abundancy of the network states delineated. I think it's more that she's wondering between the correspondence of the cluster with the electrophysiological activity.
So I think the best guess that we can make from the EFIS data is what I showed before with this heat map plot, where we looked into the like how well the cell type compositions of individual networks correlate with each other. And there we really saw that the correlation within one pattern, so like different networks after the same patterning show a very similar cell composition. So it's a bit tricky because we would need something like spatial transcriptomics to really map 1-to-1 as one cell of a specific type with the electrophysiological pattern. But the fact that we can really show that the cell composition is similar gives us a very good, or makes us very confident that we're actually looking at, very similar patterned cell clusters also on the RNA sequencing.
Thank you, Philip. And the next question is actually from Anonymous, so I cannot say the name, I'm sorry. Did you see any morphogen concentration dependent changes on electrophysiological characteristic of INS, I neurons? So morphogen concentration dependent changes.
Yeah, I guess the answer is we didn't do this, we didn't use different morphogen concentration on the electrophysiology profiling, so we cannot really answer this question because we don't know. I don't know, Philip, do you have?
No, exactly. It was just we had to kind of, select a few conditions where we were very confident that the cell type proportions would be different. So we just selected those, and those typically were not like gradients, but really different morphogens. So while it would be interesting to see like how well separated are those in the EFIS like feature space, we didn't do it for the study, no.
Thank you both. And I think you didn't do it for this study, but maybe if you can elaborate or if you want to elaborate on that, because another question would be, is your analysis working on a 3D iPSC culture? So yours so far was mostly on 2D culture, but eventually is applicable also for 3D samples.
Yeah, absolutely. So the EFOS pipeline is really agnostic to like which sample type you're using, as long as it's spike sortable, which so for most of the data sets I've personally worked with are, yeah, then you can basically plug and play the exact same pipeline.
Thanks a lot. And while I was scrolling through the question, I also see a question that is mostly directed to us. So I would take the chance to actually say it out loud. The best way to contact us is always contact our support team. You can directly contact if you have any questions, either your dedicated person, your account manager. If you're not sure, there is a support at maxwellbio.com. But I mean, we have your email now, so we will also make sure to follow up directly with any question. This is more a general reminder for everyone that we are here for you. So anything, any doubt, we are always very happy to help. Jumping back in the question from Niels, did you find evidence from preferred connectivity between subtypes?
So if I think I need to ask for more clarification, because so we found that the connectivity is different within the different conditions, or is the question if different subtypes that we inferred from the single cell clustering are preferentially connecting to other cell types, which would be a much more tricky analysis. But if it refers to the first condition that I mentioned, yes, we indeed found that the connectivity was very different between the two, between the several conditions. So this is what I wanted to show with this graph, where we found that the network density was much higher for one condition, and the efficiency, which is typically an indicator for a very mature network, was much higher for basically the opposite, in a way, patterning condition. So we are pretty confident that the connectivity is indeed altered by the patterning. Yes.
Awesome. And if Philip probably missed on the whatever you were actually asking, Niels, feel free to write in the chat. I'm trying to multitasking here. Also, the other question is about the data. So we have Mutiat wondering, are they all the data from a single population?
I guess we might need more information about the data. Is it referring to the single cell transcriptome data or the electrophysiology data? And what does population mean in this context?
Probably we can also go back to that later. And I think that we would be done with the question. The next one was that if the webinar is going to be recorded, yes. And the replay are going to be available on our website. We just need probably a little bit of time to upload it. But absolutely, this is going to be available. I don't see any other question here or here in the chat. So I hope that I address them all. And then before actually wishing you to have a happy rest of the day for wherever you are connected from, I just have a few more information actually to share with you. You know, we are all traveling from MaxWell Biosystems all around the world. We try to be very present, come close to you. Here are just a few postcards for the places that we recently visited. But actually, if you want to meet us in person, these are the next conferences that we're gonna attend. Actually, currently, we're also in China for the Smart Symposium. We're gonna be next week at Sen Symposium in Los Angeles and then in San Diego for SFN, as I announced before. But we also continue for our webinars, as I said. Actually, we have another one tomorrow. The one for tomorrow, and it's gonna be the first episode of a series of 3 episodes. So 1 tomorrow, 2 next week. That is dedicated training training on the MaxWell Live software together with our Dr. Silvia Ronchi, also product manager for the MaxTwo. So what I want to point out for this training session is that they are exclusive for our users. So if you are a user of our technology and you didn't sign up yet, please do so or let us know so that we can send you the link to attend this meeting. But another webinar that instead is scheduled for November 12, it's going to be open to all. And this is instead, it's going to be hosted by my colleagues, Pravina Manogaran, the team of the team leader for our scientific team, together with Dr. Annalisa Bucci from the IOB in Basel as well. And then a few more things. No, just thank you from my side. I'm done it. It was amazing to have you all attending this webinar. If you have other questions, please feel free to connect to us. And then I think that I would just want to take another chance to thank our speaker for today. Thank you, Suchan. Thank you, Philip. It's been amazing having you. Thank you so much.
Thank you so much.
Thank you so much.
Thank you. Bye-bye.
Thank you. Bye-bye.
Bye.
Bye-bye.
Bye-bye.